Hi everyone, welcome to the second video in the preliminary biology series. So during this video we'll be looking at 2 dot points from the A Local Ecosystem topic. So we're looking at the first focus area dot points and we'll be looking at the second dot point in the second column of the syllabus and the first dot point in the third column of the syllabus. So we'll begin with the second dot point in the second column, which is asking us to identify, so identify is the verb, the factors determining the distribution and abundance of a species in each environment. So when they say each environment, they're referring to aquatic and terrestrial environments, which you can see in the previous dot point. Okay, so we first want to understand what the verb identify means. So if we go to the Bostes website, the glossary of keywords, and scroll down to identify, we can see that it means to recognize and name. So what this dot point is asking us to do is to recognize and name any factors which can affect the distribution and abundance of species in aquatic and terrestrial environments. So now that we know what the word identify means, we'll define the other keywords in this dot point. So distribution. Now we did cover this term in the last video, but we'll define it again. So distribution can be defined as the area where an organism is found. Okay, so the key word is the area where an organism can be found. So for example, we could say that plants such as cushion plants are distributed in alpine areas. Okay, so this is an alpine area. In desert areas, there's rattlesnakes, which are distributed throughout the area. And also on rocky seashores, there's a distribution of starfish, for example. Okay, so the next word that we'll look at is abundance. Now, this is a new word that we haven't addressed in any of the videos so far. So, abundance. Now, this is basically the number of individuals of a particular species in an area. Okay, so it's the number of individuals in an area. Distribution refers to the area which the species can be found in, and abundance is how many of those species or how many of those individual organisms are in that area. Now, with abundance, we're counting the number of individual organisms within a species group. So what do we mean by a species? Well, the organisms within a species have common characteristics. The individual organisms can interbreed with one another, okay, and they can produce fertile offspring. So an example of a group of species which you could count the abundance of would be us, so humans. Now, the scientific name for humans is Homo sapiens. So humans are a species because together we have common characteristics. So hair on our head, we've got 2 eyes, 5 fingers on each hand, and we can interbreed with one another and produce fertile offspring. Okay, so we're a species group. Another example of a species is the Australian brush turkey. So with abundance, we're counting the number of individual organisms within a species group. So for example, we could count the number of individual humans within a particular area, Or we could find the abundance of brush turkeys by counting how many individual brush turkeys are within the area. Okay, so moving back to the dot point, it is asking us to look at factors which can affect where organisms live, so their distribution, and factors which can affect how many organisms are in the environment, so their abundance. So we'll break up the factors into biotic and abiotic factors. So thinking back to our last video, we know that biotic refers to living things. So let's think about what living things can affect the distribution and abundance of species. So food supply is one biotic factor. So how does food supply affect the distribution and abundance of species? Well, if you have an area that has a large supply of food for a particular species, then this might cause more of those organisms to move into the area. So this is affecting the abundance of that species in the area. Or for example, if an area doesn't provide enough food supply for a particular species, you may have some of those species or some of those organisms dying or moving out of the area. So again, this is affecting their distribution and their abundance. So if we use koalas as an example, they're usually found in eucalypt forests. So for example, in southeast Queensland and in the southeast part of South Australia. So the abundance of koalas in these areas is usually quite high because the eucalypt forests provide a large supply of eucalypt leaves, which the koalas eat. Okay, another biotic factor to look at are the number of competitors in an area. So if you have an area where there are a large number of organisms competing, so, competing for the same food source or competing for space and shelter, you might have organisms moving out of that area. Or on the other side, if there's an area which doesn't have many competitors, there may be the abundance of a particular species might increase in that area. Okay, the number of predators in an area can also affect the distribution and abundance of species. So again, if you have a large number of predators in an area, you may have organisms moving out of that area or organisms dying, so affecting their abundance. Okay, the number of mates in an area can also affect the abundance and distribution of species. So if you have a large number of mates in an area, this will allow the organisms to reproduce and thus increase their abundance in that area. Okay, and lastly, birth and death rates affect the abundance and distribution of species. Okay, so they were some of the biotic factors which can affect species distribution and abundance. We'll now move on to look at some of the abiotic factors. Okay, so the non-living factors which can affect distribution and abundance of species. So the first one we will look at is the amount and strength of wind and rainfall in an area. So for example, if we look at the dandelion, so dandelions are usually found in high wind areas. Now the reason for that is because the wind allows their seeds to spread across large distances, so depending on the strength of the wind. Okay, so this is why these flowers benefit in high wind areas. Okay, their seeds are able to be dispersed, and this will affect their abundance and their distribution. The next abiotic factor is temperature. So plants and animals will be distributed in areas where they can withstand the temperature range in that area. So some species might function better in environments where there's a narrow temperature range, so there's not much fluctuation in terms of the temperature in that area. Whereas other organisms and species might be able to live in areas where there are large fluctuations in temperature. So for example, species that live in the desert. Tides, currents, and waves are also abiotic factors which can affect species distribution and abundance. So for example, sea organisms such as sea urchins, periwinkles, starfish, Neptune's necklace seaweed, these organisms are usually distributed in high abundance along the seashore. Now, a reason for this could be because these organisms can withstand and are not harmed by the pounding of waves on their habitat. So also the waves may carry food and nutrients to the seashore, which will benefit these organisms and help them to survive. Okay, the next abiotic factors are space and shelter in an area. So in an environment, there may not be enough space and shelter for some species to survive in. So this might cause some of the species to move out of the area, which affects their distribution, or you might have some of those organisms dying within that area, which affects their abundance. The final abiotic factors we will look at are oxygen, water, and light availability. So oxygen is necessary for organisms to survive. So species will be found in areas where there's a plentiful supply of oxygen, which enables them to function. So water is also necessary for survival. So the availability of water in a particular area will affect which organisms choose to live in that area. And the availability of light is very important for plants, as light enables the plants to undergo the process of photosynthesis, which is where the plants use the light to make their food source, glucose. Okay, so that's the abiotic and biotic factors which can affect species distribution and abundance. We will now move on to the next stop point. So process and analyze information obtained from a variety of sampling studies to justify the use of different sampling techniques to make population estimates when total counts cannot be performed. So what this dot point is asking us to do is to look at different sampling techniques that can be used to measure the abundance. So techniques that can be used to estimate the size of a population. Now, an important verb is being used in this dot point, justify. So what does justify mean? Well, if we scroll down on the Bostis website, justify means to support an argument or conclusion. So what this means is we need to look at the sampling techniques and justify their use. So support why these techniques should be used. So for example, one reason why we might use these techniques to make estimates of a population is because organisms move around. So it's very difficult to count all the individual organisms in an environment if they're constantly moving around. Okay, and if population numbers are constantly changing in an environment. So these sampling techniques can be used to make an estimate. Okay, now time and cost also provides support for these sampling techniques. So it is much less timely and less costly to sample a smaller area and make estimates of the population in that area, rather than trying to make population estimates in a larger area. Okay, now these sampling techniques are also very valuable tools for conservation management of threatened species. So what do I mean by this? Well, instead of trying to capture and count all the organisms of a particular species, so for example of a threatened species in an environment, by taking a sample from that population and using sampling techniques to make estimates of their population in the environment, it limits the disruption to that threatened species. Okay, so we'll now look at some of these sampling techniques. So firstly, we'll investigate sampling techniques to measure abundance. So one method that can be used is called a quadrat. So this is what we call a quadrat. So it's a frame which could be made of wire, wood, or plastic, and it's dropped onto the ground at random throughout an area that you're studying. So let's say we want to measure the abundance of this particular green plant in this area. So the quadrat would be dropped at random in this area. So the blue boxes that I'm drawing are representing the quadrat being dropped a number of times in this area. Now, the more drops that occur, the better the results. So we use a formula to calculate the estimated population. So firstly, we need to find the average count per quadrat. So what we mean by this is you would then go and count how many of those green plants are in each of those quadrats and then calculate the average. So for example, if we look at the first quadrat, there's one plant. The second quadrat, there's another plant. The third quadrat, there's no plants. Now in the fourth quadrat, there's only less than half the plant in there. So you would actually count that as 0. So you only count the organism if there's half or more of it present in the quadrat. Okay, so in the fourth quadrat, there's no plants, and in the fifth quadrat, there's one plant. So you would then add all those numbers together and divide by 5 because you've thrown the quadrat 5 times. So 1 plus 1 plus 0 plus 0 plus 1. So 3 divided by 5 to get your average count per quadrat. Okay, so the next part of the formula is to times this by your study area. So for example, let's say that the height is 8 meters and the width is 8 meters. So the study area would be 8 times 8, which is 64. So that would go in your study area section of the formula. Okay, then you divide your study area by the quadrat area. So you can have quadrats of different sizes, but let's, for example, say that this quadrat is 1 meter by 1 meter. So the area would be 1. So 1 times 1 is 1. So that goes into your quadrat area. Okay, now there are some limitations to using quadrats. So they can't be used for organisms that are constantly moving around. So they're mostly effective for counting the abundance of plants and organisms that are immobile. Okay, another technique which can be used to estimate the abundance is a technique which we call capture-recapture. So with this method, unlike the quadrat technique, this can be used for animals which are mobile, okay, or secretive and hard to find animals. So the first step of this technique is to capture a sample of a particular animal in an environment. So for this example, we'll pretend we want to estimate the abundance of a particular bee population in an environment. So we would capture a sample of that bee population. Okay, the second step would be to mark or to tag those captured organisms. Okay, so we would mark and tag the bees that we've caught. So I'll colour in these bees to represent that we've marked them or tagged them. Okay, the next step is actually to then release all the organisms that you've captured. So in this example, we would release all these marked and tagged bees back into the same environment where they were initially captured. Okay, so that's step 3. So then you would wait a certain amount of time And step 4 is actually then to go and capture another sample of that bee population from the same environment. And step 5 would then be to count all the marked and tagged bees from your second captured population. So from this example, you can see that we've got one of those bees coloured in. So you've got a total of one of that population of bees which you had already captured the first time. Now, after performing these steps, there's a formula to be used to calculate the estimated population. So in this case, the estimated population of bees from this area. So the formula is estimated population is equal to the number of bees in your first capture. So in this case, it would be 2, times by the number of bees in your second capture. So the total number of bees, which would be 3. And then divide all of this by the number of marked bees in your second capture. So your recaptured bees, which in this case would be 1. So 1 goes here. And then to calculate the estimated population, it would be 2 times 3 divided by 1, which is 6. Now, to improve the reliability of the results, you would actually perform a few recaptures and then calculate an average value. Okay, so the quadrats and the capture-recapture method are techniques which are used to measure the abundance of organisms, so the number of organisms. We'll now look at a technique which can be used to measure or to estimate the distribution of species, so where species are found. So one technique that can be used is what we call a transect. So what is a transect? Well, this is an example of a transect. So basically the idea is to have a line which cuts right across the area being studied. So you can see in the middle here there's this piece of string, and the line could be made of anything. So it could be string, or it could be, for example, those marker sticks which can be hammered into the soil. So the study is done by moving along the line and basically noting and recording which species are located at each point. So for the example that I've drawn below, you can see that there's a marker found at 1 meter points across the area being studied. So you would walk across that line and draw any of these species that are found at particular points along the transect. So for example, if walking along, you might see a particular type of grass species that you want to study, so you draw that there. At the 1 meter mark, there may be a certain plant, a tree that you would like to study, so you draw that one. And you go all the way to the end of the transect, drawing the species that you're studying in that particular area. So this allows you to see where that species is distributed, so where it's found in that area. Now, often plants are the main subjects of a transect study because many animals move around so fast and so quickly that they could not be studied using a transect. Okay, so this concludes our second preliminary biology video. The third video will cover the next dot point on photosynthesis and respiration.